Hello again. So now I'm going to give a presentation on studies that are specific to WHICAP. So this is one of the data sets that we'll be using this week. So first, I do want to acknowledge the WHICAP investigators and WHICAP funding. This is not an exhaustive list, so there are many other researchers who have made major contributions to WHICAP, but just for space, I'm including those who really led the studies that I'll be talking about today. So we're going to have a data summary later today. So Dr. Manley is going to give an overview of this cohort. So this is just a preliminary introduction. So WHICAP is a community-based longitudinal study of cognitive aging and dementia in northern Manhattan. So it's a regional cohort of older people who are living around the Columbia University Medical Center in northern Manhattan. So this is the catchment area for WHICAP, which is made up of West Harlem, Washington Heights, and Inwood. So this study's been going on since 1992. These older adults are followed up approximately every 2 years, either at Columbia University Medical Center or in their homes. This is a racially and ethically diverse sample, so it's made up largely of these three groups, and it's quite representative of the surrounding neighborhoods in this region of Manhattan. So with this talk, I have two major goals. The first is to follow up on Dan's great talk about the residual method for quantifying cognitive reserve. So I'm going to talk about a couple of studies that we did in WHICAP to replicate and extend that model to a longitudinal framework. And then the second goal is going to be kind of like a data blitz. So I'm going to present a series of one-slide snapshots of a bunch of different studies that have looked at different variables in WHICAP that are purported to measure or to build cognitive reserve. And they examined how those variables relate to cognitive trajectories. But as you'll see, most of them do not incorporate brain data. So I think this is an opportunity here at the meeting to kind of get inspired about what we can do incorporating all of the brain and cognitive data in WHICAP. Okay, so Dan just showed this model, which is decomposing memory variance into components that are explained by brain variables and demographic variables, as well as the residual that we'd like to learn more about. So when this paper came out, I thought it was very interesting, and Dan had published the code in an appendix of the paper, and we had all of these variables readily available in WHICAP, so why not replicate it? And the WHICAP imaging subsample that we had access to at this time was pretty similar to the UC Davis sample. So it had educational and racial and ethnic diversity, and for these initial scans, the MRI data were even processed at the lab at UC Davis. So there was a lot of concordance between these samples. For example, in terms of manual tracing of the hippocampus, it was the same lab that did this in both studies. So this is probably a good test bed to replicate as a first step. Now, one important difference between the WHICAP imaging sample and the UC Davis imaging sample is that dementia was an exclusion criterion in WHICAP. So they tried not to scan people with dementia. But we still had quite a bit of memory variance left to decompose. So, almost a quarter of the sample met criteria for Peterson criteria for MCI at the time. Okay, so here's the model once again. As a result of our not including individuals with dementia, not surprisingly, these brain variables, which are purportedly measuring brain disease, explained less of the variance in memory. So in our sample, we found that these three brain variables explained a much smaller proportion of non-error memory variance compared to the UC Davis sample, which had almost a quarter individuals with dementia. The demographic variables explain slightly less variance than in the UC Davis sample. And so as a result, we had a lot more non-error memory variance that was left unexplained. So that's an important difference between the UC Davis sample and the WHICAP sample, which sort of gets at this idea that it's really hard to measure reserve in people who don't yet have brain disease. We were also, with this study, interested in figuring out where age might fit into this model. So in the original paper, age was left out because they had done previous work showing that the effects of age on memory were entirely mediated by the brain variables. But that wasn't the case in WHICAP. So we still had strong age effects on memory performance, even after accounting for all of these brain variables. Although of these three memory components, the brain component was the one that was most strongly correlated with age. So going forward with our validation steps, replicating Dan's study, we included age as a covariate. So remember from Dan's talk that the next step in this decomposition model is to see how these different variance components predict outcomes. And we tried to model that paper as closely as we could. So I'm going to show some slides that are very similar to Dan's. So here I'm showing the results from 5 different models. So each of these rows represents a different model. And these are standardized estimates from those models that looked at independent associations between each of these 3 variance components and the outcome on the left. So as you can see, our results were very much in line with the UC Davis results. So in our sample, having higher residual memory variance was associated with a lower likelihood of meeting concurrent criteria for MCI. Having more residual memory variance was also associated with higher reading ability, lower dementia conversion over the next 3 to 4 years of follow-up, better cognition in a non-memory domain. So in WHICAP, the non-memory domain we focused on was language, which, as you'll see in the data summary later, in WHICAP, the language domain is made up of a variety of language tests, from very basic tests like repetition to more complex tasks like verbal abstraction. And finally, having higher values of this residual variable were also associated with less decline in that non-memory domain over the next 3 years. Now remember from Dan's talk that perhaps the strongest test of the cognitive reserve hypothesis is that this residual should moderate the association between brain and cognitive change. So again, showing you a slide very similar to what Dan showed. Again, we have sort of brain integrity on the x-axis and longitudinal cognitive decline. This is decline in language on the y-axis. And people who had the lowest values of that residual variable had the strongest association between brain integrity and cognition, whereas people with the highest levels of that brain variable did not show much of an association at all between brain integrity and cognition. So that was pretty much just a replication of what Dan just showed. We had a little bit of longitudinal MRI data in WHICAP, so we thought we would try to extend this to a longitudinal framework. Here's a snapshot of the longitudinal sample. So this is a subset of that original sample I just showed you that came in for another MRI, I think about 4 years later. And not surprisingly, they're doing a little better than the sample as a whole in terms of brain variables, cognition. I should also mention that the quantification of these brain variables changed during this time. So now we're extracting these variables from FreeSurfer's longitudinal pipeline. We also are using a different algorithm for labeling white matter hyperintensities that was developed by Adam Brickman, which I believe has a lower threshold for labeling white matter hyperintensity. So these numbers look a little different from the ones on the previous slide. So because we only had 2 time points of MRI data, we unfortunately weren't able to model slope of brain change. We are now obtaining 3 measures of brain data in the WHICAP sample, so that's very exciting. But at the time of this paper, we just had 2 time points available to us. So we used a really simple computational framework for the study. Basically, the first step was using just the baseline data. We did a regression. So we regressed our episodic memory composite onto demographics and brain variables and called the residual our residual reserve index. And then as a second step, we took the regression coefficients from that baseline regression and applied them to MRI data obtained at the second MRI to compute predicted memory scores. So based on what your brain looked like at time 2, what should your memory score be? And then we computed the difference between that predicted memory score and what they actually obtained on memory testing at the visit of the second visit. And so now we have 2 variables representing reserve index at 2 periods of time, and at each time point, this reserve index essentially captures the difference between predicted and actual memory performance. Okay, so I think a picture is a lot easier to convey sort of what was calculated and to show you some different scenarios of people that we saw in our data. So here I'm going to show you 3 actual groups of WHICAP participants and how they looked on all of these variables. So here in red, I'm showing you their predicted memory scores based on the brain measures and the demographic measures. So as you can see, all of these groups, it's going down over time. So those brain variables are getting worse over time. And then in black, I'm showing you their actual memory performance. And here's where you see some variability across groups. So some people are showing memory decline that's out of proportion to their brain decline. Other people are showing memory decline that's pretty much in line with their brain decline. It's the middle panel. And then in the last panel, you can see that some people are maintaining memory performance or even getting better from time 1 to time 2 despite their advancing brain pathology. So overlaying now the residual, so that's essentially the difference between these predicted and actual at each time point. You can see that some people, their residual went down. So here in this first panel, at the first time point, these individuals were sort of outperforming their MRIs, and then at the follow-up, they're underperforming, so their residual declined. Other people did not show much change at all in their residual, so the difference between predicted and actual was pretty much the same at the 2 time points. And then in the third panel, you can see that some people's residual was going up. So here at baseline, this group was underperforming, and then at follow-up, they're doing better than predicted based on their MRI. So these 3 groups actually correspond to tertiles in this sample defined by the residual change variable. So in this sample, 1/3 of people showed declining residuals on average, 1/3 of people showed stable residuals on average, and 1/3 of people showed slightly increasing residuals on average. Now this residual change variable could be driven by both brain changes and memory changes because it's the difference between brain and predicted memory and actual memory. But interestingly, these 3 subgroups, who again are defined just on the basis of how much residual change they exhibited, there were no significant differences in any of the brain change variables. So all 3 of these groups showed equivalent gray matter atrophy, equivalent hippocampal atrophy, and equivalent progression of white matter hyperintensities. It's only their memory, sort of how their memory was reacting to those changes, that differed across groups. So we gave these groups some labels. So here, this first panel, we suggested that they were showing a depletion of their cognitive reserve because they had it at baseline and they lost it at follow-up. Here, these people are showing stable reserve, so they're maintaining their difference, more or less. And some people showed increasing reserve or revealed reserve because they were switching the direction of their residual variable. Now here I'm showing you a graph that is now collapsed across the whole sample, so putting those 3 tertiles together into one sample. And I'm showing you changes from time 1 to time 2 in all of the relevant variables of interest. And we use latent difference scores to compute both group level changes, so average changes in each of these variables in the group, as well as individual differences in the magnitude of that change. And what we found is that there were significant group changes in all of these variables. So first, they showed slight increase, but statistically significant increase in white matter hyperintensities, a slight but statistically significant decrease in gray matter volume, a larger and significant decrease in hippocampal volume, and then declines in both language and memory. So here right in the middle is the residual variable. So you can see in the sample as a whole, they're showing significant decline in the residual variable. So in the sample as a whole, there's some depletion of cognitive reserve, but remember there's quite a bit of variability in that change, as we saw in the previous slide, with some people showing depletion, some people showing stability, and some people showing increasing or revealed reserve. So we did some validation work to try to see if this change in residual was meaningful. So for example, we showed that changes in this residual variable predicted dementia conversion independent of baseline values of the residual, controlling for age as well. And recall that maybe the strongest test of the reserve hypothesis is that these residual variables should moderate brain cognition associations. So here we used multiple group modeling to show that those people who showed depleted cognitive reserve showed a stronger negative association between the progression of white matter hyperintensities and decline in a non-memory domain, language decline. So people who had depleting memory residuals were showing a stronger coupling of their white matter hyperintensity progression and their language decline compared to people who had stable residuals who didn't show an association between advancing white matter hyperintensities and language change. Interestingly, there was no significant difference between this stable group and the increasing group in terms of this correlation between brain change and language change. So I'm going to sort of transition now into that data blitz portion, but I thought it might be worthwhile to take questions about that stuff now before we kind of forget it. So would anyone like to ask a question about that? Yeah, please, Alden.
Yeah. So in your cartoon where you had the... Oh. It's on. It's on. So in your cartoon earlier, where you had the three graphs of memory performance versus the brain variables up a little bit, was it intentional that the high memory performers are declining faster and low memory performers are increasing, or was this a cartoon?
No, this is actual data.
Okay.
Yep. And the three groups were created just based on the magnitude of their residual change. And so we took those three groups who were defined based on their residual change and then looked at their changes on all of the other variables. And we ran regressions to see what variables predicted membership in these groups. So how these three groups differed. And they did not differ in any of the longitudinal brain measures, but they did differ in so this group did show lower baseline brain, so specifically white matter hyperintensity. So they had more white matter hyperintensities at baseline, even though they showed no differences in the rate of change in white matter hyperintensities. And they also showed lower memory at baseline. So you can kind of see that, so the red slopes are all the same. So these three groups had equivalent brain change. This red line is a little lower. It's kind of downshifted compared to these two groups, and that's because they had more white matter hyperintensities at baseline. And then you can see the memory scores, they had lower memory scores at baseline, whereas these two groups were not different in their baseline memory scores. Yeah. Does that help?
Yeah.
Yeah. I really like this picture. I feel like it answers every question. Well, we'll see.
Annie Christine, hi. So this is sort of a question both for this cohort and UC Davis and sort of the cognitive reserve studies in general. So one of the main hypotheses that you guys have put forth is sort of trying to look at the coupling between cognition and brain based off of this cognitive reserve measure. But I'm a little confused about how to look at that coupling when brain variables are used in creating your cognitive reserve measure. So whenever you're then looking at that coupling, is your sort of brain measure going to be independent? Are those different measures, or is that really the MEM-B? And if so, how circular is that?
Yeah, that's a great question. We're very concerned about circularity in these models. So we have done a good job, I think, of talking about non-memory domains as the validation cognitive domain. But you're right that the brain variable we're using is usually the same brain variable that we're using to compute the residual. So one answer to that is in this particular example where I showed that these two groups differed in their coupling. They actually did not differ in any of their brain variables. So they both had the same baseline brain, and they both had the same change in their brain. So these groups didn't differ in their brain. They only differed in how those brain changes related to changes in a non-memory domain. So, I think some of these analyses get around the circularity, but not all of them. So I think this is a really good question to keep in mind as we work with these models. Thanks.
And another thing is that we're looking at the effects of the brain reserve index on future cognitive decline too. and defining it differently. So that helps to get around that issue.
Okay, so that was helpful. Okay, so I'm really going to shift now and kind of go into this data blitz. So most of these studies I was not involved in. Some of them I was, but I wanted to show a bunch of these studies. First, to preview the types of variables that are available in WHICAP, since this is one of the data sets that some of the work groups will be working with this week. So this kind of gives you a preview in advance of Dr. Manley's data summary later today. And the other point is just to kind of give you some context and background as you plan your papers and your proposals with WHICAP data, kind of what's already been done and how can you show that you're building off of that previous work. Okay, so this is a very early study. So this came out just shortly after WHICAP was initially funded. Here I'm showing survival analyses looking at the main and interactive effects of education and occupation on dementia conversion. And basically, this just communicates that this data set has been used to answer questions about protective factors in dementia for almost 25 years. And one of the first studies to show a lot of these effects. This is a more recent study looking at education in WHICAP. So I talked a little bit about this in response to Gerald's question after the last talk. So with this study, we were interested in following up on some conflicting findings in the literature about whether education moderates the rate of cognitive decline. So there's a ton of evidence that there are strong level effects of education, that higher education is associated with higher cognitive level, but whether education additionally moderates the subsequent rate of cognitive change has been pretty conflicting. And we had reason to believe from a paper that came out of the IELSA, the Integrated Analyses of Longitudinal Studies of Aging, that the effect of education on slope, on the rate of cognitive change, might differ depending on the distribution of education in your samples. So specifically comparing samples, more highly educated samples, where each additional year of education really just corresponds to more college, and lower educated samples where each additional year of education corresponds to an additional year of primary school. So we broke the WHICAP sample up into low education, high education groups, and then looked at education as a continuous variable within each of those subgroups, how it predicted cognitive trajectories. So we used a pretty sensitive curve of factors model. I should have shown our fancy structural equation modeling here, but I think we did a really good job of isolating error variance. So we had really a sensitive way of testing for these education effects on slope. And so we found very small but statistically significant effects of education on rate of cognitive change in both of these groups. So not only was there a level difference in each group, there was a very slight slope difference, linear slope difference, in that people with more education were showing less decline. As you can see in the low education group, that kind of petered off with the quadratic part of the model. And with regard to downstream mechanisms, we also looked at income as a potential downstream effect of educational experiences. And we found that in the high education group, income explained the protective effect of education on cognitive change, but not in the low education group. Income did not explain the protective effect of additional years of primary school. So this kind of gave us some initial evidence that maybe the mechanisms underlying the protective effects of education differ, whether you're talking about elementary school or secondary and post-secondary school. So Jennifer Manley has looked extensively. Oh, yeah, question?
Back one slide.
Yes.
It looks like the 9-year group is below the 8-year group on the left. Is that odd?
Oh, yeah, that's a good question. I believe that that these are not comparable, that there was some within-group centering, but I'm not sure. So I'll check on that. That's a good observation.
And those are T-scores, not necessarily raw scores.
Yes, these are T-scores, right? Yeah.
The lower group each at baseline.
Yeah, I think that's what it is. Thanks. Okay, so reading level now. So Jennifer Manley has done a lot of work on reading level, which appears to be a better proxy for educational experiences in non-Hispanic Black older adults who were more likely to attend schools characterized by shorter school years and higher student-teacher ratios, fewer resources, et cetera. And so she has shown that independent of years of education, higher reading level is associated not only with higher cognitive level, but also less cognitive decline. And reading level helps to explain Black-white inequalities in cognitive aging above and beyond years of education. So this is a more recent paper coming out of the Psychometrics Conference where they sort of more thoroughly operationalized some of these constructs of early life educational quality and later life literacy. And this study was cool because it really took advantage of all of the extensive questionnaire data in WHICAP as well as objective testing, and I think this was administrative data. So a good example of the data that are available in WHICAP and how we can use it creatively. I showed you this study in my last talk. So this was the WHICAP study that was interested in looking at associations between education, reading level, and pathology, white matter hyperintensities, controlling for cognition. So recall the results of this study were that at any given level of language or speed performance, those with higher education or higher reading level had more pathology. So they were able to maintain their particular level of cognitive performance despite their having more pathology. So this is really one of the few studies that I'll present now that incorporated brain data into this examination of cognitive reserve proxies and cognition. So Nick Skarmeus in WHICAP has done a little bit of work looking at leisure activities. So this is a checklist of hobbies and recreational activities. And this paper showed that older adults in WHICAP who reported engaging in more activities at baseline were less likely to go on to develop incident dementia. For simplicity, I'm just showing you that this is a middle age group here, but the curves looked very different, very similar for the younger and older groups as well. And here, these curves were created just by a simple median split on the activities questionnaire. So this is a questionnaire with 13 activities, and they just took a median split, people who reported 6 or fewer versus more than 6. And they also did a little bit of work to break down that activities questionnaire and look at individual items. And so this effect seemed to be driven by specific items like reading and visiting friends and family. So some preliminary suggestion that the types of activities people are engaging in matters for dementia risk. We've also looked at bilingualism in WHICAP, specifically within a subgroup of WHICAP participants who are Spanish-speaking. So this paper was just on individuals who were Hispanic immigrants and who were all tested in Spanish. So they were primarily Spanish-speaking. So we operationalized bilingualism as proficiency in English. And as you can see, we did see a bilingual advantage in initial performance on tests of verbal and nonverbal abstraction and letter fluency, but there was no significant difference in subsequent rate of cognitive decline or in dementia conversion. So I think Dr. Arce and others are doing some work to follow up on this finding in WHICAP. So I mentioned that WHICAP is a regional sample, so all of these individuals are living in northern Manhattan, but there is quite a bit of variability in region of origin. So particularly in the Hispanic subgroup, many of those individuals were not born in the United States. And within the non-Hispanic Black subgroup, most of those individuals were born in the U.S. South, so not in New York, where they are currently residing. So this was a study that tested whether state of birth, so focusing just on U.S.-born participants in WHICAP, whether state of birth explained cognitive variance in non-Hispanic Black and white WHICAP participants. And as you can see in the whole sample, so all of the U.S.-born participants, state of birth explained 7 to 14% of the variance in cognition. So this is another finding that can be followed up in I don't remember the number of the work group, but one of our work groups is interested in this environmental context question. 4. Work group 4, plug for work group 4. We also have a psychosocial work group. So I think that work group might be interested in how psychosocial variables can deplete reserve or contribute to reserve. This study was looking at depressive symptoms in a slightly different way. So it was testing whether these sort of common reserve proxies moderate the known negative association between depressive symptoms as a risk factor and cognition. And I think the findings of this study were contrary to their expectations. So they found that people with more education, so the dotted line here, they showed a stronger negative association between depressive symptoms and cognition. So it appears that having depressive symptoms might sort of overshadow the protective effect of education and also reading level. But this is cross-sectional data, so it's not clear sort of what the direction of the effect is here. Interestingly, this study, they found the opposite results when they swapped out this outcome of cognition for a brain outcome. So when they looked at how education and reading moderate the association between depressive symptoms and total brain volume, they found that people with higher reading level showed less of a negative association between depressive symptoms and brain volume. So that's more in line with the brain resilience idea, that these proxies are buffering the negative impact of depressive symptoms on brain structure. But again, this is a cross-sectional study, so definitely a lot of room for longitudinal follow-up. Okay, so just to summarize some of the studies that I talked about today. So first, our replication of the residual reserve index work from Dan's group provided additional evidence that this might be a useful method of quantifying and studying cognitive reserve. Our longitudinal extension provided evidence that this residual reserve variable changes over time, and that those changes differ across people and are meaningful because they predict important outcomes above and beyond cross-sectional measures of the residual reserve index. So it's possible that this might be a method of tracking the depletion of cognitive reserve, so tracking this dynamic construct of how memory reacts to changes in brain structure. And then finally, my whirlwind tour of cognitive reserve proxy studies in WHICAP suggests that all of these variables seem to be relevant to our understanding of individual differences in cognitive function. But as you recall, very few of them incorporated brain variables. So I think additional studies are needed to use all of the brain and cognitive data to really understand how these factors relate not only to cognitive functioning, but the concept of cognitive reserve in particular. So hopefully I got us back on track a little bit with time. But I have time for questions. Thank you. Paul, that's the best idea you've ever had.
Laura, thanks for that nice overview. I really like the data blitz idea. So on your second study that you presented, where you're looking at low and high education groups, my question is about the distribution of ethnicity. So in the Dominican Republic, until very recently, education levels were the average level was primary school. So I'm wondering, do you still see variation in terms of ethnicity in these two groups?
There is some variation. So we have decent representation of education, particularly in the earlier cohorts of WHICAP. So even in the white sample, a lot of those whites are themselves immigrants, so people who are fleeing World War II and whose educational experiences were interrupted by the war. So in that early group, we do have diversity in education, even within groups. That being said, there are very clear differences in the proportion of those three racial ethnic groups in each of these samples. So there are many more Caribbean Hispanics in the low education group. So we statistically controlled for that in these models, and I think we did have enough variance to do that. But I don't think we did a good enough job in this paper testing for interactions. And so I think that could be a really cool extension. Shout out to work group 2.
And then my second question is, you mentioned that many of the respondents were born in the South and migrated.
Yeah, 45%, or I think 55% of the non-Hispanic Black subgroup in that study were born in the U.S. South. Yeah.
And so based on the timing of migration, was that from the Great Migration?
Yes, yeah, yep. Yeah, I don't think we had, for that group, age of migration. So we do have age of migration for the non-U.S. born sample, but not the U.S. born sample. So we did not have age of migration, which is an interesting idea. One thing that Sam did with that paper, as I recall, is she also did some sensitivity analyses about movers versus stayers, as I recall. So trying to get, because they also had HRS data in this paper. So trying to break down, is it just a south-north difference, or is it a migration, non-migration difference? I don't remember all the details of those analyses, but I know that they were interested in that as well, but not age of migration. Yeah. Great questions.
That was just wonderful, Laura, and I want to ask a provocative question. Oh, I'm sorry. I'm David Johnson from UC Davis. I'm a new Californian, formerly of University of Kansas, and I'm a neuropsychologist. And love and very interested in this topic for all of my career. I want to ask the question, I think we're limited by the temporal resolution of the data, but the provocative question is, why, instead of looking at this as a moderator effect, why isn't this actually a full or a partial mediator where, building on the previous questions about the security that we have as A-beta as a signal of the disease or the neuropath or the brain measures, why isn't this actually a better measure of the neuropathological disease, and we're missing it in some other litmus that we have within the MRI or the PET scan? And if so, then this isn't a moderating vehicle at all. It's actually a partial or full mediator. And of course, only longitudinal mediation would be able to tell you that with 3 or more observations. But I just wanted to offer that, especially on some of your more provocative slides about this switching interaction. That's much more emblematic of a mediator than it is a moderator. And just wanted to know some of your thoughts, because maybe that's a different frame to look at the data.
Yeah, I really like that question, because I think it brings up testable hypotheses. It's some people talk about cognition as a biomarker for disease, and that's really not how we've been talking about it here. So we've really been coming at it from what's been written about cognition for decades, and that literature would suggest that no matter how good we are at measuring these structural brain variables, there should always be residual variance, and people can always deal with that pathology. But it's an empirical question. Thanks.
Thanks, Laura. That was great. I'm Maria Cleamore from UCSF, the shy member of the organizing committee. I had 2 questions. One is just a confirmation, because I've never seen it juxtaposed. I think that you said that state of birth explains 7 to 9% of the variance in the cognitive measures, and that you also said that the brain MRI measures explain 7 to 9% of the cognition measures. Is that correct? Did I hear that wrong?
Yeah. So
it's just a remarkable.
Yes. The 3 brain variables, explained 7% of the non-error memory variance, and state of birth explains 7 to 14% of the cognitive variance. You're right. Coincidence?
This is a kind of incoherent question, but I'm hoping you can just sort of explore this. I'm really troubled by the role of measurement error in a lot of the cognitive reserve approaches, and even the picture that you showed that says it answers all questions. And I don't remember which picture that was, but it seemed to me like that was consistent with regression to the mean, and regression to the mean was going to play a huge role in this. But maybe you have methods to account for that. So if you could talk about, if you're worried about that, what you're doing to account for it, and what you think we need to do to understand the role of that.
Yeah, absolutely. So I'll say 2 things about it. One is, so the latent variable version of the residual model that Dan presented and that we originally replicated, that does attempt to partial out error variance, so it's using known reliabilities of the measures. But our longitudinal extension, we didn't use that latent variable method. We used just straight up regression. So our residual was absolutely more confounded by error than the latent variable method. So hopefully in the conference going forward, I think the longitudinal group, shout out to work group 5, is going to be using a latent variable model to regress slopes and to separate out, hopefully, error variance better. So that's a huge difference in problem with our extension compared to the original latent variable work. And then for regression to the mean, so there are equations that compute how much regression to the mean you should expect, and that's based on sort of the test-retest of your baseline and follow-up. So if you look at test-retest of the baseline and follow-up residual, we computed that we would expect, I think, 10% of the follow-up variance in the depleting group to be explained by regression to the mean, and then 20% of the follow-up to be explained by regression to the mean in the increasing group, which is the group we were most worried about. That's the weirdest group. So regression to the mean, we think, probably explains about 20% of that increase that we see, but that's a relatively small proportion of the variance. So I absolutely agree that regression to the mean is a problem and needs to be accounted for, but I don't think that all that change can be explained by regression to the mean. But again, we should be testing that and measuring it as we go forward. So that's a great question.